Yeah, so why do we bother doing solution NMR essentially is where I was getting to. And fortunately, a solution NMR is good for a few things. So flexible proteins, which is something we're gonna see tomorrow, I think, with something that solution NMR is pretty good to. It's the go-to technique for these proteins. Lowly populated or excited states, which is something I'll be talking about later in these talks. A weak binding, whether that's weak, that cannot be detected by any other technique. That's also something that NMR can do. But what about large assemblies, which is the topic of this section? And since I'm first, I'm supposed to give a little bit of background, I think, to this topic. So this is where NMR gets into problems, solution NMR gets into problems, because as you can see here, there is molecular weight limitation. I'm showing some examples of small unfolded residue peptide. You can see all these lines are pretty narrow. You get a lot of signal. For a small folded protein, then you get a bit more peaks. The lines get a bit broader, but still it's pretty nice. There's this 70 residue protein. But if you go larger, you can see here that all these lines are very broad, and you can actually see only in this case, you can only the signal comes from a little bit of unfolded regions in this part of the protein. And people have tried to solve this, and one way to solve this is by deuterating your protein. So here at the bottom, you have a medium-sized 30 kilodalton protein, which is protonated. And this spectrum basically is a classic fingerprint HSQC spectrum that connects the amide nitrogen to amide protons. And you can see here, this part of the spectrum is pretty busy. And because the line width is kind of large, then all the peaks end up on top of each other, and we can't really get much information out of this. But if you essentially deuterate all the non-exchangeable protons and record the same spectrum, this gets much better. Now you can see the line widths are a lot smaller, and you can actually separate most of these peaks in this case. So this works for medium-sized proteins, and people have taken that step further. So you can, in the background of these highly deuterated proteins, you can essentially reintroduce protons that have favorable relaxation properties, and those are methyl groups in this case. And then that allows characterization of much larger assemblies, and this is now standard. A lot of people do that if you want to look into some larger proteins. And I'm just showing an example here from our own work with Christos Pliotas from Leeds, where we're looking at membrane protein embedded in nanodiscs. So the complex is large, 200, maybe closer to 300 kilodaltons in this case. But as you can see here, you can nicely see at least these methyl groups at the top of the protein. Although this is the way that you can characterize directly, you can actually directly see NMR signals from large assemblies in solution NMR. Today I'll be talking about something that's a little bit different, and basically using chemical exchange NMR methods that are designed to look at chemical exchange phenomena and how this can be used to look at larger complexes. So chemical exchange is shown here. You have a protein that exchanges between state A and state B with a forward and a backward rate. And these states have different chemical shifts in this case. So this is the chemical shift of state A, and that's the chemical shift of state B. And this example here is just a simulated 1D spectrum where these two states are populated 50-50, which is kind of unusual. It doesn't really happen most of the times. It's more common that your excited state B is much less populated. So if you drop the population down to 10%, you get to this situation. If you make the exchange go a bit faster, you can actually lose the peak of the state B peak. So state B may or may not be observable directly in your spectra. But the good thing is that as you can see here, we can see differences in what's going on with the peak of state A. So the question is, can we just use that information, which we'll always have, in order to characterize what's going on here? And these techniques have been very popular in the last 10-15 years by many big people in the field: Louis Kay, Marius, Art Palmer, or Peter Wright, many others that I don't mention. But a lot of these techniques are kind of designed to look at a case like this. So when you have a monomer exchanging with another monomeric state. So what I'll spend the first part of the talk is to see what happens and how these techniques get affected if the state B is not a monomer, so it's a larger oligomer, which means the R2 of this species is much larger than the R2 of the main state. So if you have a scenario like this, if you have some chemical exchange, probably the first thing that makes sense is to do some sort of titration. So you're pushing the equilibrium from state A to state B, and then you see what happens. And this is what I've plotted here. So this is just again the simulated 1D spectra as I increase the concentration or the population of state B. And here we have the state B is lowly populated and we're intermediate exchange, so you don't actually see the peak that corresponds to that state, which would be here. But instead, as you increase, as you push the equilibrium this way, you just see that the peak of the main state moves that way, right? And then it gets broader as well. And then you can quantify that. The people call that exchange-induced shift. So you can quantify how far the peak from state A has moved in this plot here as a function of the concentration or the population of state B. And you see you can get a curve that goes up to 40 Hertz in this case. Now, what happens if our excited state is not a monomer? It's a larger oligomer here, which I've shown like that. It's probably not very nice, but so everything else is the same. And as you can see here, even if you look at the spectra, so this is what we had before. Now this peak of the main state doesn't really move, and you can quantify that in this plot here. So the exchange-induced shift is much smaller. It goes up to 15 Hertz only. And actually, there's an expression from Skrinnekon-Fekete from this paper about the value that gives the value of this exchange-induced shift. And you can see here in the denominator there is this delta 2 component which is basically the difference in the R2 states between states A and states B. And essentially that quenches the value of delta X if you have exchange with a large species. Then the next thing I want to talk about, and we're going to see that later in the example I'm going to give in the second part of the talk, is this technique called CPMG, which has been very popular essentially in the last 15 years. And Luis K has a lot of reviews about this, so you can read that if you really want to know what's going on there. But essentially, when you have your signal in the XY plane, you apply your focusing pulses at a different rate. So if you don't have many of these refocusing pulses, there's enough time in between these pulses for this exchange to happen. But if you increase the frequency of these pulses, this time becomes shorter, and there's less chance that you're going to get this going on in between the pulses. And then you can quantify your peak intensities here. And if you have a change in the right time scale, you get this curve that looks like that. If nothing is going on, you would get a flat curve down here probably. So this technique kind of relies on the fact that these state A and state B have different chemical shifts. So it wouldn't work if these two states have exactly the same shift. So we can do again a little bit of simulation. If we have a monomeric state exchanging with another excited state that's also monomeric, with this exchange parameters, we get a curve looks like this. So you get this REX term, which is essentially how big the curve is from down there to up here, which is about 10 per second, for example. But if we actually, again, look what's going on, if state B is not a monomer, so it's bigger, you will see that the curve here has moved up to reflect the larger R2 of the second state. But also this REX term is much smaller, so you don't really get much of a difference from the first to the last point. And again, the same paper from Skrinnekon-Fekete have an expression for the REX terms, and you can see delta 2 again in the denominator. So again, in this case, the fact that our state B is a large oligomer kind of quenches what we see in our experiments. And the last experiment I will talk about is the off-resonant R1-Rho experiment, which was Al Palmer has done a lot of work on that. And again, if you have a monomeric sensing with another excited state that's a monomer that has a different chemical shifts, you get like this profile that looks upward, if you will. But recently, work from the K lab has realized that the same experiment, if your excited state is a large assembly, then this curve is actually inverted. So the fact that you get a curve that looks like that is indication that your direct indication that your state B is a large oligomer. And we're going to see an example of that later. So just to summarize this part, so I just put together all these plots I've shown in the previous slides. Green is the monomer, if the excited state is a monomer, and the gray curves is if the excited state is a large oligomer. And you can see all these three different experiments, they behave differently. So the relaxation properties of the excited states influence what we see in the NMR experiments, and some of them give very characteristic profiles for low and high molecular weight excited states. So this is the kind of the introduction part, which is like lays the foundations of what I'm going to talk about in the second slide. So you can, if you want to read more, of course, Louis K has a lot of reviews about this exchange. Vitali has a very nice review in JBNMR in 2019 that deals with exactly what I was talking about. If your excited state is a large oligomer, which is a nice trait, and we put a few things together and recently in this paper more towards large chaperone complexes. So now if we move to the second part, I'm basically going to use these three techniques that I mentioned in the introduction part and apply them to our favorite chaperone molecule, and which is called DNAB6. This is work described in these 4 papers. I'm not going to talk about all of 4 of them. I'm just going to summarize the bits that are relevant to the introduction part. So this is my chaperone slide. What basically trying to answer what is a chaperone, and essentially chaperones are a network actually of proteins that promote protein folding. And you can see that here, if you have a protein that emerges from the ribosome, which is nicely shown here in this schematic, you can have this chaperone molecule, which is shown here in blue, that binds to it and promotes its folding to the native state. It can recognize some other chaperones recognize folding intermediates and prevent them to misfold. But if they do, sometimes that happens, then there's other sets of chaperones that bind to those and label them for degradation. And even if these misfolded substrates, they form these large aggregates, whether those are amorphous aggregates or amyloid fibrils, there's another set of chaperones that can even disaggregate these. And what is good about these chaperones and their complexes is that they're very, very dynamic. So on their own, the chaperones tend to be very dynamic molecules, but also when they're bound to their substrates, so which is good for NMR, because I don't think TRIM can do that yet. So we can use the methods I described in the beginning to actually characterize this, what's going on with these proteins. So and I'm not going to talk a lot about the chaperones and what they do, but I think we're going to hear more and better introduction later on. So DNase-AB6, this is the molecule we like. So this is an HSP40 chaperone, and these proteins are regulators of the HSP70 cycle, which is very important. I'm not going to talk about it, but we will hear about that later. But the DNase-AB6 has other functions that they were identified recently by the work from the Kapinga lab in Netherlands, which are not dependent on HSP70. And these are basically related to inhibition of protein polymerization reactions. So you can see that here in vitro, for example, you have A-beta aggregated on its own. So this is a measure of how fast the protein aggregates. So if it's on its own, it aggregates pretty quickly. Then you add a little bit of the DNA JB6 there, and you see that in substoichiometric ratios that prolongs the aggregation time course of the peptide. And you can do the same experiment in vivo actually. So you can see here this is GFP tagged A beta, and you can see that on its own it forms this punctal fluorescence, which means that the protein aggregates inside the cells. But when you express DNA JB6, you don't see that anymore. You do see this diffuse fluorescence, which indicates that the A beta is now monomeric. So DNA JB6 has a highly substoichiometric activity. And this protein inhibition functions, they kind of like encoded in these two domains. So the DNA JB6 has 4 different domains, these folded domains in the JD in the N-terminus and this C-terminal domain. And in the middle, you have these 2 highly disordered domains. But it seems to be that for the inhibition function, this ST and CTD domain seems to be important. We'll see that later. And also it's important because it's implicated in various diseases, myopathies, breast cancer, virus polymerization, or even neurodegeneration diseases. Okay, so we wanted to look at this protein, but we very quickly realized that and that's was something that was actually known before, that it forms very large oligomers that are actually important for the function. So the functional form of the protein is the oligomeric form. And here you can see a profile, a DLS profile. You get a nice peak around 24 nanometers. In cryo-EM, you see a mess, essentially, like a lot of oligomers of various sizes. And you can put those down in a cell filtration column. They come all in the void, but there is some monomer here. So there seems to be some exchange between the oligomeric forms and the monomeric form. And you can take an NMR spectrum of this, which looks like this. But it wasn't really good for us because solution NMR is not probably the best thing to look at a sample like that. So we had to find a way to make the protein behave better. So the first thing is to delete, that we tried is to delete this low complexity domain right in front of the C-terminal domain. So this is creating this construct called delta ST, which now is monomeric. And you can see the spectrum of this truncated variant overlays with the full-length protein. So that's good. It doesn't seem to, the deletion of this part of the protein doesn't disrupts the oligomers, but it doesn't affect the structure of the monomer, which is good. And actually the same thing happens for if you have only the C-terminal domain. So this is again monomeric, and the peaks of this construct overlay with the peaks of the full-length protein. So that was good. And we end up doing the NMR structure of this construct here, this delta ST construct, which I'm showing here. As you can see, it's a very flexible protein. At least 50% of it is disordered, and so you can see here in red the J domain, which is alpha helical, and then this domain, this GF domain, which is actually functionally important. It forms this little helix in the middle of it. But I'm not going to talk about this. It's a topic for a different talk. And that leads to the C-terminal domain, which is beta-stranded. And you can see that it flops around. It doesn't really have a preferred orientation between this. There isn't a preferred orientation between these 2 domains. And these are the PDB IDs for the monomer. And we put together, recently we submitted an entry in the protein ensemble database that kind of tries to show the relative, when these 2 domains come together, how they do that. So you can look at that up if you want. But the point is, can we say anything? This is good. We can tell about the monomer quite a bit. But can we say anything about the oligomers, which is actually the functional form of the protein? So if you do something like that, the first thing is do titration experiment, as I said before. So even just looking at the spectra, so if you have this protein at low concentration, this is 200 micromolar, it basically looks much better than what it looks like at 1.1 millimolar. And you can see some peaks are more affected than others. and they all kind of are in the C-terminal domain. And you can actually quantify this in this plot here. So this is the R2 of the difference in R2 between 600 micromolar and 400 micromolar. So if there wasn't any formation of any big stuff, this plot would be around 0. The fact that we see a significant difference in the R2 of these two samples that immediately suggests that there is something bigger forming in there. And you can see this is whatever is going on is larger for the C-terminal domain. So this suggests that the C-terminal domain is in some ways involved in this oligomeric species. So maybe the C-terminal domain is the core of this oligomer, whatever oligomer forms, and the rest is kind of tumbling on its own. So basically, what I'm saying is maybe not clear, but that's probably makes it more clear. What I think is going on in this case is that in the wild-type protein, we have an equilibrium between monomer and oligomers. But that's pushed all the way to the right, so this is towards the oligomeric side. But if we have this construct, this truncated constructs, the delta ST construct or the C-terminal domain on its own, this equilibrium still exists, but now it's pushed on to the left. So we have a mainly monomeric sample, but we still get some of the oligomers that we're interested in. And then we did the CPMG experiments I talked to you about in the previous slides. This is the CPMG experiment at low concentration, so relatively low, 120 micromolar, and these curves are flat, so nothing really interesting is going on there. But if we increase the concentration to 500 micromolar, we start seeing curves. So there is some exchange going on, which is concentration dependent. Suggesting of oligomerization again, and you can see where these exchange events are taking place, and they're essentially clustered around this N-terminal part of the B1 strand, and maybe at the C-terminal strand as well. And you can actually measure this the difference in chemical shifts as a function of concentration. So this is the exchange-induced delta X plots that I've shown in the beginning of the talk, and you can do that for H alpha protons, C alpha, and C beta carbons, and you can see that you do get a curve. So it's kind of large. It goes up to 20 hertz or 40 hertz for some other ones, and just by looking at that and the fact that we do get large CPMG profiles, then you know that there has to be an exchange with a state that has similar properties, relaxation properties with the monomer. Otherwise, these values will be quenched, and you wouldn't actually get that. And you can see here that for the most part, there is a linear dependence of the values of this exchange-induced shifts on protein concentration, which is going to be important. And the other thing you can say from this is that because we get a positive H alpha and C beta shift and a negative C alpha shift, we know that whatever is going on suggests that the excited state we get a more regular beta strand configuration just by looking at the values of those. And then we did the other set of experiments, this off resonance Unrun row experiment. And you can see here that the profile is inverted. And what I've shown you in the introductions to the talk, that the fact that you do get inverted profiles is directly indicative that there is something bigger, a state with a large R2 in the samples, and you can actually quantify how this is formed. So this is this delta R2 that I showed between only two concentrations in the previous slide, but now we just plot as a function of concentration, and you can see that this is clearly not linear. It's actually a second order dependence on protein concentration. So these two slides, the previous slide, the fact that we get large CPMG profiles and delta values of exchange-induced shift suggests that we have a small exchange with something that's small. This slide suggests that we have a change with something that's big. So this is direct evidence, and the fact that this one is linear, the other one is not linear, this is direct evidence that whatever is going on there is not only two states. We have at least two states in this case. And actually, you can play some games with the methyl groups. So here, this is just a spectrum, a carbon spectrum of the methyl group. You get four lines depending on how many protons are up or down. And these proton transitions, they basically are reflected in this energy diagram. And you can see some of them have slow relaxation properties. Some of them have fast relaxation properties. And if you remember again, fast relaxation is bad, especially if we have a change with large species. And indeed, if you do the CPMG experiment on this system, so this single quantum carbon CPMG, you get curves that are not very informative. They're, for the most part, they're relatively flat. But if you do Vitaly's magic, so he selects only these two transitions in these methyl groups, which are slow relaxing, and that's described in his paper from 2020. Now you can see that the curves are much better. They are bigger, and we can actually use them. We can actually analyze them. And if you do that, basically, you find out that the methyl groups in the C-terminal domain, which are shown here, they get restricted in the oligomer. So we take all the information together. We put this scheme. We put together this scheme that describes what's going on in this equilibria. And basically, what you have here is a main state, which is monomer, that exchanges with a dimer, which this B1 strand is involved in this dimerization. You get 7% of this at 500 micromolar, and then 3%. This dimer essentially interacts with something a lot bigger, which is 3% at this concentration. And you can get the rate of formation of these things. They're in the millisecond range. This step is twice as slow as this step. And the nice thing is that from the analysis, you can actually get the size of this species, which ends up to be around 20 to 25 subunits. So it's pretty big. And just to relate that to the first part of the talk, so this exchange, this first step is kind of probed by the CPMG experiments and this exchange induced shifts. So you get a linear dependence because this is a dimerization, and that the size of this dimer is not hugely different than the size of the monomer, so it doesn't quench your CPMG or delta X values. And this second step is probed by the fact that we have these inverted profiles in the operational Saint-Laurent row, and this nonlinear dependence on concentration as well. But why do we actually care about this? You may ask, which is a good question. I think that this actually is quite important to understand how this protein works. And if we take a step back and see how a protein may aggregate. So here we have a monomer, then through nucleated polymerization, it forms this nucleus, and then you have another conformational chains to create these fibrils. And if you then look at what chaperones that inhibit these sort of reactions do, they tend to be unstable on their own, and that's actually the case for our molecule, DNA JB6, the Bricker's domain, AB crystalline, HSP27. All of these, they tend to form some pool of heterogeneous oligomers, which are shown here. So you have different states of these chaperones. They can exist as oligomers of various sizes, and these are not static. They change, they're dynamic, so they change their subunit with one another. And during that subunit exchange, you can imagine that these states, these oligomers, can capture some of these species here, and they wouldn't allow them to further aggregate to this fibril structure. So it's important, essentially, what I'm saying is important to understand what's going on here in the subunit exchange in order to understand how these chaperones may inhibit the aggregation of their substrate. And I put here and we do have some ideas based on the schema I showed you in the previous slide that how that may happen for DNA JB6 now. But if you do understand that, then we should be able to modulate and change what's going on in this equilibria. And the first thing we did, so we have to remember that I was only using truncation constructs so far. So what happens in the full-length protein? So we basically took the full-length protein and we decided, which has this serine threonine domain, which was deleted in the previous constructs, and we just then decided to delete the last bit of the C-terminal domain, which is shown here, which is actually disordered, and see what happens. And as you can see here, maybe a lot of peaks are the same between these two constructs, but a lot of them in this delta C10 construct, which is this guy here, a lot of the peaks are missing, and all the missing peaks are essentially in the C-terminal domain, which means that the deletion of the last 10 residues has destabilized the structure of this. We don't actually see that in the spectra, but the rest of the protein is fine. So what happens in terms of oligomerization? This is just a delta filtration profile, and you can see that delta DNA JB6 is an oligomer. This delta ST, as shown before, is a monomer. But also now, in the absence of the C-terminal domain, which is not properly folded, this construct is also monomeric. So which basically means that in order to get efficient oligomerization for this protein, we need both this serine threonine domain and the C-terminal domain. And more recently, which is described in this paper for a couple of months ago, we decided to make a mutation right here. So this is in the B1 strand, which is involved in dimerization. And I've shown you this scheme before for the wild type protein. And if you repeat everything for the mutant, then you basically see that the population of the dimer is now reduced twofold. So from 7% at 500 micromolar, it goes down to 3.5%. The population of the oligomer is also down to 0.5% only, which actually proves that this dimer is on pathway to oligomerization. But this mutation has also increased the population of this partially folded state on this side, which forms very fast on the microseconds time scale, while this is happening on the millisecond time scale. Okay, so conclusions. For the first part, I think you should be careful to take into account the size of the species in exchange when even interpreting simple NMR experiments, because that will change what you see. And NMR relaxation-based experiments can inform on large excited states. So I've shown you that we can actually use these experiments to detect the presence of large species. We can get their populations, the rate of formation, but even their size. And in terms of the what's going on with our chaperone, DNA JB6 chaperone, it looks like both the C-terminal domain and this low complexity serine threonine domain are responsible for the oligomerization of this protein. We have some suggestions about the mechanisms of subunit exchange within these oligomers, which is something that we're actually testing now. And I forgot to say that the mutation that I talked about in the B1 strand is actually important for the disease, Parkinson's disease, because it's lowering the population of the functional oligomer. So then the protein is not able to do its inhibition of the alpha-synuclein in the cells. So that's my last slide. So yeah, so some of the work I've mentioned was done at NIH when I was at the lab of Marius, and now in the Asprey Center, I would like to thank Emma, Billy, Christos, and Arnaud. I think that's a good place to announce that we actually be moving to Imperial College in October. So if anyone has known anyone that wants a postdoc, then please get in contact. We'll be very happy to hear from you.